Hey, so for this video, I wanted to start getting into some of the virology, which I think is probably what most people are interested in. And I'll have a lot of images here. I'm just going to talk through some of the basics on virus structure and some of the nomenclature. And then we can move forward into infection in the next videos. As far as a definition, what is a virus? I think this definition, I mean, you could look up a definition of viruses and you might get something different. This is my definition of a virus, which I've kind of compiled from, a couple of years now of looking at viruses in undergrad and talking about them. And I think this encapsulates some of the important components of what makes a virus a virus. So viruses are obligate intracellular parasites consisting of a protein coat surrounding a genome, which is made up of either RNA or DNA, and they require the host metabolism to complete their life cycle. So I just want to break down what that means a little bit. Obligate intracellular, meaning it has to be inside of a cell. It doesn't live on its own outside of a cell, and if it is outside of a cell, it's metabolically inert. It consists of a protein coat surrounding a genome of either RNA or DNA. We know that the protein that surrounds the virus is called capsid protein, and all viruses have that. And they encapsulate either an RNA or a DNA genome. So thinking back to the previous videos I made describing the differences of what RNA and DNA are, you can be one or the other if you're a virus. But never both as far as we've ever found. I think the other really interesting thing here is that no other organism on Earth can be an RNA genome. Viruses are the only things that do that. I guess getting into viroids too, but that's a different topic. But viruses and virus-like things are the only organisms that have RNA genomes. And then the last bit here requires host metabolism. This gets back to the parasitic nature of what a virus is. A parasite is something that has to live on or in a host organism, and it benefits off of it in a one-way street. The organism doesn't benefit from that virus. The virus benefits from the organism, and that's parasitism. And so I think that's a pretty thorough definition, but you may see others out there. I think also that the interaction between the host and the virus, the host meaning the human or the animal or the plant, the interaction between those is not quite an arms race like we think of with bacteria, but maybe more of like a game of chess or a dance in where one player does one thing and very systematically and very deliberately the other player does something that will negate that prior move from the other organism. And so all successful pathogens have to find some way to antagonize those host defenses. The host, evolves some defense, and if it's a successful pathogen, it will evolve a defense around or an antagonistic mechanism to get rid of that defense. And it's a back and forth. Just a couple little tidbits here about viruses. They infect every known living organism on this planet. Every class of organism carries viruses. They're not inherently bad. There's many viruses that we're infected with from early in age, and they have no deleterious effects on us. In fact, some of them even promote life in some ways. There is a virus called syncytia virus, which is the sole reason why mammals have a placenta. It's because of that virus. That virus endogenized into those mammalian lines, and it allowed for the formation of a placenta because of some cytopathic effect that the virus had, this creation of a syncytia. So I think it's really interesting. We probably wouldn't be here without viruses. And then another, just maybe it's a soapbox point, but they're not strictly alive. And I think I've mentioned this before. Viruses they're not live, they're not dead. I like to think of them as either intact or degraded. You could say that you killed a virus, but really you're degrading that virus. It wasn't alive in the first place. There's another sort of point made by Vincent Racconello, who is a virology professor, is pretty well known. He thinks of viruses as this two-stage cycle. Outside of the host, they're not alive at all, but inside of the host, they carry out metabolic functions, and therefore he considers them alive in that sense. It's just more of a way of thinking of it. And just to put things in perspective, we're talking about these particles which are on the nanoscale meter, and that's 10 to the minus ninth. And if you don't know what that or have a grasp of what that is, here's that numeric value typed out. And so it's that's 10 to the ninth, so of a meter. That's pretty small. And because of that, we can't resolve these with traditional light microscopes. We have to use other methods typically because they're just too small. There are some that are on the micrometer scale, and from there you can resolve them with a light microscope, but they're few and far between. They're only the largest viruses that can be visualized that way. So to give you an idea of some of the complex morphology of viruses, when I say morphology, I mean the shape of viruses. Here's just a sample of the thousands of viruses out there, and I think one thing you can take away from this, though, is they fall into three broad categories. We call them icosahedral, or helical, or everything else gets grouped into complex. And so these are your icosahedral ones are these spherical looking capsids. All of these, like your influenza, we would consider these icosahedral, meaning I think it's like a 20-sided, 20-faceted, I guess I don't remember exactly why. It has to do with the number, though. That's where that name comes from. But at any rate, you get a spherical capsid. Helical, we get things like what's presented here for Ebola. They have these filamentous, sort of fibrous looking capsids to them. So tobacco mosaic virus is another one which infects plants. And then everything else, like rabies virus, actually is considered complex. It's not helical, it's not icosahedral. And then bacteriophages, which are definitely complex. As I mentioned, these are made up of the genetic materials made up of either DNA or RNA surrounded by protein. So although this cartoon doesn't show it, these are all proteins on the outside, and they're surrounding the genetic material. So just to finish this then with talking about the three different morphologies in depth, to give you an idea, because you will hear this on the news or in popular press. So this is first looking at the icosahedral capsid. In this case, it has a lipid membrane around it, which they're calling an envelope. And then there's proteins which are stuck into that envelope. These proteins are what bind to host cells, like the spike protein. So with the SARS coronavirus 2, the spike protein that they've mentioned is ACE2. I'm sorry, not the spike protein is the viral spike protein, and the receptor is ACE2. So the spike protein on the virus is binding to our host cell, ACE2. These surround a layer of protein in here called matrix protein, and then within that, we have our genome stored in here. Upon binding, this will fuse with the host cell and release this genome into the cell. Looking at a helical example, all animal helical viruses have a host-derived lipid membrane. They don't have to have that in plant viruses, but in animals that seems to hold true. So this is an example of Marburg or Ebola virus. This could depict either of them. But we see here that the protein, which was just studying that outside of the virus in the last example, is here as well. We have these viral proteins on the outside. And then the capsid proteins are all these right here. And these are coiled up and surrounding in this helical fashion the genome of the virus, in this case, the RNA genome. And then it's packaged with the machinery that it needs to execute once it gets into the cell. And then last, I think these are maybe the coolest looking ones, are the complex viruses. And for this example, I've just chosen to look at bacteriophages because I think they're the most interesting and alien looking viruses out there. So we see here on the background, this is the capsid, and it has this stalk right here, and then these these feet, these spike proteins here. Well, these aren't spike proteins, but they have these little spiky legs. And it sort of looks like a lander or something, a moon lander, but they're really neat looking. And the cartoon here shows, oh, there we go, the names, the capsid, that sheath, and then, yeah, so tail fibers is what they call them. And then in there would be the spikes. And the spikes are what penetrate into the host cell and then allow the genome to be injected, or ejected rather, from the head down the sheath and into the cell. And so just in case you think that you don't believe this, or you don't believe me, or whatever the case may be, we've actually visualized these with, I say we as in like the scientific community, has visualized these via electron microscopy. And so we actually have seen what these look like under an electron microscope, and that is exactly what they look like. So I think that's really cool, and sort of allows you to get an appreciation for the diverse range of viruses and the way that they could look as far as their morphology goes. Last thing I want to talk about here then is the genomes, which I've mentioned on a number of occasions. The important points here are that all viruses have to find a way to get to mRNA. So they start off with some genomic material, whether it's DNA, double-stranded DNA, double-stranded RNA, whatever it is, and it has to get to mRNA. If you think back to the transcription translation videos, you would understand that. That's because you don't make protein unless you have mRNA. So if a virus wants to replicate and make more of itself, it needs to first take its genome and synthesize it in such a fashion or an orientation that it can make mRNA to make protein and then reproduce itself. And so these are grouped here. This is called the Baltimore classification, and it's just a simple way to think about the starting genetic material and what you have to do to get to DNA or to the mRNA. So if you start with DNA, you can immediately start synthesizing through transcription mRNAs. If you came in as a single-stranded DNA, you would have to make that complementary strand of DNA first. So this would have to be made double-stranded, and then it could synthesize mRNAs, because that's how transcription works. If you came in as a double-stranded RNA virus, you have a negative strand and a positive strand. And so what could happen is either the positive strand is immediately translated, so it's opened up and translated, but more than likely, I think what's happening in as far as what I've been taught is that the negative strand gets made into a positive strand mRNA right here, and then you make proteins from that. Same thing works for a virus coming in as negative stranded RNA. It gets made into a positive strand RNA, and that can get translated. And then, so I'm going to skip the RNA one. That's pretty obvious. It comes in as plus RNA. It can be made into protein right away. And then this one right here, this class 6, is how retroviruses work, like HIV. And I think this one's probably more complex and maybe one of the more interesting ones. This comes in as RNA, and through a DNA intermediate, because it has reverse transcriptase and it can make DNA. That goes against the dogma of biology. If you remember, I said in a couple videos back that the central dogma of biology is DNA to RNA to protein. Well, in this case, we're going backwards. We're going RNA to DNA and then to RNA and protein. So it's really interesting. Comes in as RNA, it makes a DNA copy of itself, then it makes a double-stranded DNA copy of itself. This will go into the host chromosome. It'll get into the nucleus, and it'll integrate into the host chromosome. And that is why you have HIV for life. It becomes part of your actual genetic material. And from there, it can put out these mRNAs, which get translated into proteins, and it can also make new positive-stranded full-length genomes to infect new people. So it's just kind of an interesting, I think it's a really interesting lifestyle for that virus. 2 other points here. RNA viruses, when I think of RNA viruses, I tend to think of there being a lot of viruses that cause disease in animals and specifically in humans. And this SARS-CoV-2 is just another example of that. It's a positive stranded RNA virus. And these viruses are notorious for high mutation rates and being able to escape some of the immune system checks and balances that are there. So just an interesting thing about them. So with that, I think I'm going to leave it here. And on the next video, I will start talking a little bit more about what occurs during those primary infections and some of the first steps. So thanks for listening.